Yes, hello everybody. My name is Heike Rebholz, and I'm very, very happy that I'm allowed to present our work and to talk to you today about the work we're doing on OCNDS. I'm sharing my screen. Okay, so I work at the Institute of Psychiatry and Neuroscience in Paris, and in our lab, we're studying mainly mouse models with the aim to better understand OCNDS. We're not studying a single mouse model, but we have different mouse models that we want to compare, simply because the condition itself is not defined by one single variant, not just one mutation that all the patients share. But as you know, we have different and a big variety of variants of mutations that also generate a spectrum of symptoms in the patient. So in a way, we kind of do not want to reduce our research onto studying one mouse model, but we want to widen it to be really more in the realistic context of seeing the whole spectrum of symptoms and maybe also understanding a spectrum of mechanisms that underlie the symptoms. This is the main reason why we have chosen to compare different mouse models. So first, a little bit of a background on CK2, in case you need a refresher on protein biochemistry. So CK2, as you know, is a protein. So it has DNA that encodes for it, like every protein has a blueprint in the format of DNA, and this is where a mutation occurs. But then that DNA is translated into a protein, and the protein is a three-dimensional chain composed of things called amino acids that basically perform a variety of jobs in our cells, in all of our cells, and in our bodies. And CK2 is not only a protein, but it's also an enzyme, which means it's a protein that has a job to do, an activity to perform. And the activity of CK2 is to be a kinase, as the name says. It's called CK2 for casein kinase 2. So what kind of activity is that kinase activity? Well, it means, we have to move that. It means that a kinase takes a group, a molecular group that is called a phosphate, and transfers it onto another protein that we call now substrate, and modifies it thereby. So we have a substrate that becomes a phosphorylated substrate, which will actually impact on the ability of that substrate protein to do its own job or to interact with other proteins within the cell. And so CK2 is basically that entity that mediates that phosphorylation onto many other proteins. And so this process can be also reversed. So there are phosphatases that take off the phosphate from the substrate again, which allows the cells to really respond to demands, for example, of a nutritional state or a growth state or any sort of signaling that is needed to perform within the cell. That can be basically, it's a on and off switch, that phosphorylation, for a variety of things. And so CK2 is one of many kinases that we have, but it's an important kinase because it's expressed in all of our cells in our body. And it's also conserved, which means CK2 is also present in other living organisms, in mice, but also in organisms as small as yeast, yeah, and organisms as small as a worm or Drosophila, a fly. So CK2 is highly conserved, which means, yes, it's present in many different organisms, and that's always an indication that it's an important protein, an important kinase. So we know it's expressed in every cell, we know it's expressed in many organisms, and we know it has roles in many functions that a cell has to assume. It's important for cell growth, proliferation. It has roles in cancer, in creating cell shape and movement, in cell-to-cell communication, and many more. We also know that this kinase is very active. That means it is doing that job of phosphorylating a substrate very readily. It doesn't need to be pushed into doing the job, but it's basically constitutively active. As soon as it recognizes a substrate protein and it has an available phosphate to transfer, it will do that. And in addition, it has a lot of substrates. So it's not a kinase that just phosphorylates a handful of substrates, but because the recognition motif for CK2 is not very stringent, that means that we have a lot of proteins in the proteome of a cell, within the cell, that are potentially phosphorylated by CK2. Just all of that to tell you that this kinase that we're looking at is an important kinase, and this is just an example to illustrate that, whereby we see with how many proteins in the cell, and this is probably just a fraction thereof, CK2 can functionally interact. So you see that that network here with CK2 in the middle, and all these proteins and hubs of proteins are actually somehow related to CK2 and its phosphorylation activity. Also, it's expressed in every organ and in every cell of our bodies, and this just shows you that it has a high potential of phosphorylating many substrates, many proteins in our cells. So CK2, we're talking here about OCNDS. So the main culprit, or the main organ that we're interested in when we try to understand what is going on in OCS is obviously the brain. And so we studied a while ago already the expression pattern of CK2, the various subunits thereof in the mouse brain, and we could show, and other people have done the same, that again, CK2 is really expressed in every region of our brain, and most probably in all of our brain cells. Here's a structure that was solved by Dr. Neefend a while ago that shows you that CK2 is quite a complex molecule because it has actually 4 different units that have to come together to form what we call holoenzyme, the full enzyme. We have 2 catalytic subunits and 2 regulatory subunits that form this, what we call butterfly structure of CK2. And once that is put together, CK2 is ready to go and to do its job of phosphorylating substrates. And we know in the brain there are many substrates that are of crucial importance. You know, they are to do with, you know, Parkinson's disease, for example, with Alzheimer's disease. We know a lot of molecules that are known there to be, you know, involved in pathology are also CK2 substrates, but that doesn't mean that OCNDS has something to do or has a comorbidity with any of these proteins that are playing roles in these other conditions, since we really don't know what the real function of these proteins is in the brains, and we just know that there is a correlation with certain conditions, but that really doesn't mean that OCNDS in any way is related to, you know, any other disease outcome than OCNDS. So I don't want to confuse you. I was just trying to say that many substrates that we know are important in brain physiology, you know, happen to be CK2 substrates. So just again, CK2 is important in the brain. And many people, including us, we have studied that CK2 plays a role in modulating or in being active at the synapse. And the synapse in the brain is the point where 2 nerve cells, 2 neurons interact and communicate with each other. Here's a little scheme that shows you the cell that releases a neurotransmitter, so releases the message to the receiving cell, which we call postsynaptic cell. And then this cell receives that chemical message and then knows how to respond to that chemical message downstream of it. And so we know that CK2 has substrates that are really very much important in mediating that communicated signal between 2 nerve cells. I'm sorry about that. For example, one receptor, it's a glutamate receptor on that side of the synapse, is phosphorylated by CK2. We know that for sure. But why do we study mice and not just brain cells or anything? Why do we want to work with mice? Well, we already knew from earlier studies that when we knock out CK2 in certain brain cells, of the mouse, we get behaviors that are reminiscent, at least in part, of behaviors that we see in patients with OCNDS. For example, that could involve the tics or the stereotypies, that could involve hyperactivity of those mice, that could involve learning and cognitive defects that we've detected in those mice, or changes in circadian rhythm that could be maybe translatable to changes in or difficulties in sleeping, and so on. So we have already had, prior to OCNDS being described, we knew that CK2 plays a role in some of these behaviors that are linked now to OCNDS. So that makes the rationale quite strong to think that mice are a good model to study OCNDS. And you have maybe seen this scheme before of the CK2 and the many variants, and by now we have more even, that are linked to the expression of OCNDS. For example, you know the 198R mutation is the one that is most commonly occurring in patients. Then we have others that are also, you know, representing a whole bunch, a whole, you know, group of patients. And then we have the more rare variants or rare mutations as well. And so we decided already a few years ago that we have to start with the most prevalent mutation, the 198R mutation, but that we would also want to study other mutations because we wanted to see over the whole length of the protein, what do different mutations engender? What do they entail in terms of the activity and the behavior of the protein in the cell? Because we might expect different things to happen where the mutation is in that part of the protein to whether the mutation is very early on at the beginning of the protein. And we tested already the activity that results when we change, you know, the sequence to that mutated sequence of the 3 chosen mutants, the 198, the 47 mutation, and the 312. We know that all the mutations will lead to a reduced activity of a CK2 when we tested basically in vitro in a tissue in a little, you know, not in a cell, but when we do that, what we call in vitro in the laboratory. So we have indications that yes, this activity of CK2 is changed when we have these mutations. And because CK2 is, as I told you, a quite potent or a quite a kinase that has many different substrates, it also makes sense to not look only at one known substrate, but to look at the whole set of proteins that are present in a cell and see whether phosphorylation of many substrate is actually altered when we, for example, inhibit the activity of CK2. And that was, I just chose that, I found that from a PhD thesis from the Litchfield group a while back. This just shows you when you inhibit biochemically, so with a drug, CK2 activity, you have a whole bunch of proteins that are altered in every cell in their phosphorylation status, showing you that really the effect of inhibiting the activity of CK2 can be, you know, can affect many, many proteins in a cell. So that was encouraging as well. So we started to test 2 of our mouse models. We used brain lysate. We used brain tissue from the mice and did the same thing. We subjected these lysates, these proteins, to an experiment called, or an analytical approach called mass spectrometry. And we found that, you know, for example, here for the 312, we found almost 2,000 proteins to be changed in the extent to which CK2 has done its job towards phosphorylating these proteins. So we see really relevant and strong changes actually in both lines. And when we look further and want to see what are all these proteins that are affected in those mutant mice, in the mouse models of OCNDS, what do they do? We find very often the majority of proteins has something to do with what I told you before, that cell-to-cell communication, that synaptic signaling between 2 neural cells, between 2 nerve cells. Here is another scheme with that cell that releases the chemical signal to the postsynaptic cell. And so this domain here, we call that the synaptic domain or the synapse. This is the region of a nerve cell that is most affected by in its phosphorylation status in those mouse models of OCNDS that we have tested so far. You see here major hits all in the synaptic compartment of neurons. And for example, this is another way to analyze the phosphoproteomic analysis from the mouse brains. There are algorithms in the web that you can use where you run your results. And again, we see this is a schematicized synaptic cartoon here. You have here the presynaptic and then the receiving, the postsynaptic cells, and everything that's either red or blue is affected in the brains of our OCNDS models. We then look more closely or in a different way. We look really using microscopy, electron microscopy, to look at the synapses, just this thing here. At this synapse, we look really with a microscope, with a very high potent microscope where we can really see individual synapses. Here you see, for example, the cell that is releasing the chemical message, and behind this double line, you can see the postsynaptic side of the membrane, of the synapse. And here again, so what we found when we quantified a lot of cells or a lot of neurons, that the one change that we find is that this connecting area, the postsynaptic density on the side of the receiving cell is changed as if it wanted to compensate for maybe reduced communication. So this is the kind of thing, I just want to give you a few examples of the type of work we're doing and kind of the beginning of the results that we're collecting from the OCNDS mouse models. We're also growing the nerve cells in dishes here, and then we image them with fluorescent molecules, again, to count whether the synapse formation is in any way altered in the mouse models of OCNDS. And indeed, we see changes when we look at that synapse formation in the tissue culture dish between the normal wild-type mouse, we call that, and between the OCNDS model mice. We also have found, with the same approach using mass spectrometry, we found that one organelle besides the synapses, which is called the mitochondria, are quite affected in one of the lines that we have studied so far. And what it means, so mitochondria are organelles in the cell. Every cell has many mitochondria. They're very important because they're what scientists call the powerhouse of the cell. This is where the cells generate a molecule that is used to generate energy, that every enzyme, every mechanism in the cell that needs energy can use that unit of energy that's produced by mitochondria if it needs energy to do something. So mitochondria are really crucial for survival of neurons, and as you maybe know, the brain is very much in demand of the energy, of our energy consumption. A lot of sugar or food that we eat will be used up as energy in our brains. And so we looked at these mitochondria in mouse models of OCNDS, and we find in one mouse model, we've only started with this, that while the number of mitochondria is not changed, the status, the health status, let's call it, of the mitochondria is altered and is maybe not as good in the mouse models than in the wild-type mice. So let's move from the brain to maybe other symptoms that you have observed in your family or in your patient that has OCNDS that are not directly brain-related or cognition-related, but maybe we call it comorbid. It also occurs. We know that just as with other neurodevelopmental disorders or autism spectrum disorders, we often find in OCNDS patients, for example, inflammation of the gut or problems with the gut or constipation or diarrhea, things like that. So we can also in the mouse models study, of course, gut morphology, because there's not so much difference in the digestive system between us and a mouse model. So this is possible to study as well using the different mouse models. We also know that the immune system is affected in many neurodevelopmental disorder, and in some of the OCNDS children. So again, we can study that in the mouse models. We know that seizure prevalence is higher, of course, in OCNDS patients than in non-OCS people. It doesn't affect all patients. We know that, of course, and you know that very well too. It might be just a percentage, maybe just a 30% that have seizures. Some have more, let's say, impactful seizures like generalized tonic-clonic seizures. Others might have more even undetected seizures that we might not be aware of. That might happen during nighttime. We don't know, but definitely the mouse model is a good model to study seizures because you can control the onset of seizures with a drug. So you can give them a drug to really induce seizures and see what happens and see if there's a seizure proneness, so more readiness if the mouse models are more easily entering a seizure than, for example, their littermates that don't have the OCNDS mutations. Then, of course, we could do cognitive tests with the mice. That is the first thing we do, and we see that we get some cognitive defects that we can find in our mice, but not all tests show that. So it's not like over the board that all cognitive tests that we perform with the mice, the OCNDS mice perform worse. That's not actually the case. But in some, and repetitively in some, we can see that they do perform differently to their littermates. And we can also, of course, investigate motor strength and motor learning. So what would be paralleled by, hypotonia or problems in achieving motor milestones such as walking, etc. Here are just examples very quickly whereby we have seen in the intestines and in the gut of mouse models changes, where, for example, the gut is an organ that needs to have a big surface, a big interaction with the nutrition, with the food that is coming through. It wants to absorb a maximum of food, and therefore we have these, villi, we have these invaginations, and we see on a morphological level that there are changes, and also in terms of the amount of mucosa producing cells, which could have something to do with generating constipation or stool of different maybe not ideal consistency, for example. So we find changes in the amount of cells that we have, also in the amount of immune cells. So all this we're in the process of quantifying. Another example is the spleen. I chose this because it's so very obvious and really easy to see that the OCNDS model mouse has an enlarged spleen size. Again, the spleen is very much part of the immune system and of the keeping the immune cells and the blood cells healthy and in the right balance. So we have aimed, or we are continuing to try and find behaviors that we can test in the mice that have relevance to OCNDS phenotypes, OCNDS symptoms. And we have tested, tests or paradigms in the field of motor defects, in the field of cognition, in the field of sociability, in the field of these tics or stereotypies, in the field of mood states, epilepsy, and so on. And depending on the different models, we don't get always overlapping phenotypes or symptoms. This is what we actually expect because that's what you see in patients too. Different patients have very different symptom profile. And so we see with our different mutations also that they don't necessarily all have the same behaviors. So what I have here marked in red are the behaviors that we find consistently across our mouse models. What I marked in blue are the ones that we find in more than one, but not in all, for example, right? And the other tests we haven't repeated yet in the mouse model. So we're really just not even a third way into what we call phenotyping the mouse lines, because it took a long time to receive them. And some we don't even have yet ready to be tested because they need to have a certain age and we need to breed a certain amount of animals first. But let me go back to the patient phenotypic or symptom spectrum. This is what many people have done, including us, just comparing the literature. And this is just a little section of mutations that shows you not even that within the 198R mutation do we have exactly, that's the most prevalent mutation, do we have exactly the same phenotypes. We have core overlapping phenotypes, of course, which is the stature, growth problems, motor, and speech development. But when, for example, when we look at seizures or sleeping problems, some have, some don't. So this is really the reality of the condition. But let me tell you that, of course, OCNDS is not different to other neurodevelopmental disorders. So other people who study other conditions have exactly the same, findings, that we cannot define OCNDS by just one thing, by one symptom profile, by saying this is how a patient must look like and not in any other way. There's always this spectrum. So this is a question that is posing itself to everybody who's researching on neurodevelopmental disorders. So it's kind of normal, and it's just it requires perseverance and work to characterize all the different phenotypes and try to understand which phenotypes are really core and which are maybe, specific to one or the other mutation, or which are really just variable and depend just on the genetic background, since every patient has different parents, right? And every patient maybe has a different ethnicity and comes from a different region of the world. So many factors come into play here. That's why a lot of people need to work and put together their work so that we can come up, hopefully in not all too long, in not too much time, with really a better understanding of, what is the core phenotype, and how can we model that in what kind of mouse model, and what are the specific phenotypes or symptoms. And this is really what is the most important, because we want to, of course, find something to help patients, right? Is it a drug or a therapy? And so we were looking for, when we have 4 mouse models, we're really looking for that really important soft spot in the middle where we can find core mechanisms that can be targeted by drugs, by medicaments, by therapies, and that all the different models can benefit. But it might turn out that it's not that easy, and that we have to subgroup different mouse models or different patient variants, and that they will then get slightly different treatments depending on, are they in the yellow subgroup or in the core subgroup, or really this is work that needs to still be established. But as a group of researchers all working together on OCNDS, hopefully we'll have answers not too far down the line from here. I want to thank people that helped and are continuing to work with me. First, I want to thank, of course, collaborators. I want to thank the OCNDS or CK2, CSNK2A1 Foundation, Gabrielle and Jennifer Sills. Very much thank you to you, and the collaborators, some of which are also part of the consortium within the CSNK2A1 Foundation. Some are in Paris and just next door, so we can really work closely. And then the team in my lab, Jose, who has done a lot of work, Dimitra as well, and Barbara and Marianne have helped as well. So thank you, and I'll be there for questions for you later on. Thank you very much.